Hi everyone, welcome to Raaonline. I'm Dr. Priyadarshini Shanmugam, and today we are going to talk about the complement system. Okay, and the learning objectives for today's session will be: what is the definition of complement? What are the general properties of complement? Okay, the components of the complement system, complement activation, the different pathways. Then what are the effects of this complement activation? So once the complement gets activated, what are the effector functions? Then complement, how it is regulated within our body. And then what are the problems that arise due to a deficiency of complement? And just before starting, I would like to tell you that it is spelled as complement with an E. Okay, and that differs from the compliment an I, which you usually give to praise people. And the term complement is actually given to a set of proteins which are present in our serum, and they complement, that means they add to the activity of our immune response. Okay, so they augment our immune response. It is like a complement. So that is why the term has been given to it. So actual definition is, it's a group of proteins normally found in the serum, but usually they are inactive. But when they get activated, they play an important role in augmenting or improving or increasing the immune response. And this serum complement constitutes about 5% of the normal serum proteins. Okay, and their level will not increase following any infection or vaccination. Like for example, suppose you get an infection, the corresponding antibodies will increase, but the complement level will be the same. Okay, and what are the general properties of complement? You need to know that all the complement components, that is the first one, that is C1 is one component, and always the function starts by means of the binding of this complement to an antibody. And you should know that the complement binds only to the Fc region of the antibody. So antibody has got a constant and a variable region. The complement will go and bind, the protein will bind only to the Fc region, that is the constant region of the antibody. Okay, and once it binds to the antibody, that term given to it is, it's either known as complement fixation or complement consumption, because once the complement binds to the antibody, it disappears from the serum, so it cannot be detected from the serum at that time. So that's why it's also called as consumption. Okay, so just know that it will bind to the Fc portion of the antibody, okay, and not to the variable portion. Second thing is, a complement, usually the protein will not go and bind to the antibody which is free. That means a predisposition for that is, the antigen should bind to the antibody. So already you should have some foreign substance in your body to which the antibody or the immunoglobulin has bound, and to such antibodies these complement proteins will go and bind. It will not go and bind to free antibodies which are already usually normally circulating in our blood. And this complement fixation is not determined by the nature of the antigen. So any kind of antigen, when it binds to the antibody, it will trigger the activation of complement. So this fixation of complement is not determined by the type of antigen, but it is determined only by the class of the antibody. Okay, the class of antibody, meaning whether it is IgG or IgM, only those two can fix the complement. Other antibodies cannot fix or activate the complement. The third important property of these complement proteins are that they are species non-specific. That means you can take complement from one species and that can go and augment the response of this antibody from another species. But the efficiency, okay, of this kind of interaction will decrease with the increase in taxonomic distance. Means if they are taxonomically totally unrelated species, the efficiency of this complement activation will be less than between related species. Another important property of this complement is that it is heat labile. So complement can get denatured when you heat the serum, the patient's serum, the person's serum, you take and heat it at 56 degrees centigrade in a water bath. You keep it for 30 minutes. The complement will lose its activity because the proteins which form the complement system get denatured at this temperature. So any serum which has been kept in 56 degrees for 30 minutes will lose its complement activity, and such serum with lost complement activity is called as inactivated serum. And regarding the components which are present, which constitute this complement system, almost 30 different serum proteins are there. Okay, they are all known as the complement components. And in addition to it, you have some regulatory proteins which are also coming as complement components, and the properdin system. So they are all given names, and how they are named? They are all given numerals as their names. So there are 9 components in complement, C1 to 9. Okay, they are numbered capital C. Capital C is the short form for complement. So C1 to 9 they are numbered. Okay, and the names are such. And C1 has got further 3 subunits, C1Q, C1R and C1S. So QRS. Okay. In addition to these proteins, you also have the properdin system. Then you have the regulatory proteins, which are all named by letter symbols. So the C1 to 9 is numerals. After that, you have the letter symbols like factor B and so on. So this picture actually shows the different components of the complement and the different types of pathways, which we will be addressing in the next few slides. Okay, so here you can see C1, C4, C2, then you have C3 here, and you have names given to factors, as I told you, no, the alphabet. So it's represented by capital B. So factor B, factor D, H, I. So all these are the names given to the properdin system, and then you have the regulatory proteins also. Okay, so all these together make up about 30 different components in the complement system, and complement is nothing but a set of serum proteins. Okay, and they get activated by certain mechanisms. The most common way of activation is an antigen going and binding to an antibody. Now coming to the synthesis of the complement components, a liver will be the main site for the synthesis of this complement. Minor sites where again the complement synthesis takes place in smaller amounts is the monocytes in the blood, the tissue macrophages, as well as the epithelial cells of the GIT, as well as the genitourinary tract.